Okay, today we're going to talk a little bit about personalized diagnostics and treatments. Our example is going to be expression profiling, which is to say, looking at the levels of specific RNAs in early breast cancer cells. So these are not a substitute for the traditional methods of looking at tumors, but they are a definitely strong supplement to it in a way that people can actually avoid chemotherapy and sometimes surgery. Okay, the idea of expression profiling. Of course, cancer cells differ from normal cells because of the mutations that deregulate cellular pathways. So what's going to happen, of course, is that the RNAs that are produced in cancer cells are going to differ from comparable normal cells. Some genes will be expressed higher than normal, some will be lower. Okay, so this whole idea of transcriptome is simply the collection or total RNA population in one cancer cell type versus another. Now, it's important to realize here that different types of cancers do have different transcription profiles, different transcriptomes. And by looking at those different types, you can predict better how the tumor is going to behave. So, examples of pathways where RNAs might be deregulated. For example, the cells should activate cell death pathways, and they don't. You can have anti-growth signaling pathways that are messed up. Growth factor expression can be messed up, and also response to nutritional signals and DNA damage, etc. So cancer cells deviate in many, many ways from normal cells, and importantly, different types of cancer cells differ from each other. Okay, so Mammaprint is a microarray, and it's used to predict breast cancer progression. Okay, it uses a signature. It doesn't do the entire transcriptome of breast cancer cells. But what it does is use 70 different genes that are involved in different types of cancer, or that can reflect the behavior of a tumor as an indication of how that particular tumor will behave. And the idea of this particular test is to look at early breast cancer to find out whether chemotherapy can safely be avoided. Okay, now microarray technology has largely been succeeded by next generation sequencing, but because it takes the FDA so long to approve things, this one is still in very widespread clinical use. So this is using expression signatures, which is to say what actual populations of RNA or what diagnostic RNAs characterize the predicted behavior of early breast cancer. So what was used in developing this particular assay, it was to look at specifically early stage breast cancer, tumors less than 5 centimeters in diameter, and that the idea is you can characterize what RNAs are there, which will characterize those which would later progress versus those which don't. That could allow some people to avoid chemotherapy. Okay, so the study was done originally using a very large number of patients or tumor samples from a very large number of patients that had had breast cancer and had had surgery or not, had had chemotherapy or not. And the idea is to compare what was known about the behavior of particular early tumors as a reverse predictive value, to give it predictive value on how similar tumors would behave. So this was based on a very large amount of data from a very large number of tumors and characterizing the expression levels of many, many RNAs in those to try to extract out those particular RNAs that might be helpful in terms of predicting the prognosis. So the idea is to try to identify low risk versus high risk patients based on what particular RNAs they have that are either being upregulated or downregulated. So this is done by a custom microarray analysis. Okay, so this is like a very giant reverse dot plot, right? So microarrays can be used at the expression levels of thousands and thousands of genes. In this particular case, we're not. We're going to be only looking at the expression levels of about 70 of them, ultimately. Okay, so the way microarrays work in general, and this is going to be an expression array versus a genotyping array, but the idea is the same. We have a fixed support, which is in this case a microscope slide, and we have probes of DNA, which are affixed to the microscope slide. Now those probes are going to be complementary to specific RNAs, and hybridizing of the RNAs to those particular probes are going to tell us about the expression level of those particular RNAs. So in this example, in this case, the target sequences are the RNA populations, which have been enzymatically converted to DNA, those and labeled with fluorescent compounds. And those target sequences are floating around in solution, and they will complement the probes on the array, and that we can then compare a reference hybridization to the target cell tumor RNA to get an idea of whether any particular RNA is up or down regulated. But the idea, of course, is the probes are the DNA, the target is the RNA essentially in solution, which is labeled. And we're going to look at the comparative hybridization of a reference versus the tumor RNA to try to figure out who's being overexpressed in which particular tumor. So here's what one looks like. Here's a microscope slide, and it has 8 little chambers in which the hybridization will occur. So inside each of those 8 little chambers are all the probes that have been fixed to the slide, actually a lot of them, not just the 70. And after hybridization, what's going to be read is a pattern of spots that reflect the expression levels, relative expression levels of all those different RNAs. So in this case, the oligos are about 60 nucleotides long, and there are about 1,900 of them actually in each one of those little wells. So you can hybridize 8 different patient samples on one slide. So each one can be used for 8. Each one has about 1,900 oligos that are specific for whatever target RNA is being measured. And so the idea, of course, is we're going to try to identify low risk versus high risk progression based on which RNAs exactly are up and low regulated. Okay, so here's a picture of what this is going to look like. Right, we have different genes, oligos specific for different genes fixed to the array. Gene 1, gene 2, gene 3, gene 4. We're going to hybridize. The green is going to be a reference RNA population, and the red is going to be our sample RNA population. And what we're simply going to do here is compare the relative expression levels of the reference versus the sample for all those, in this case, 8 genes, 7 genes, but on the array it's going to be 1,900 different spots. So you can see what's happening here. Gene 1, there's a lot more of the sample RNA hybridizing to the probes. So you've got a lot of red here hybridizing to the probe and a little bit of green. So that would represent an RNA that's overexpressed in the tumor sample versus the reference. By contrast, look at gene 3. Here we have a whole bunch of little probes. They're all the same within the spot, where we have 3 greens and 1 red. And so what that means is that the sample, which is the tumor sample, has less expression than the reference for that particular gene. So this sample, all the different spots need to be hybridized. And what happens eventually, what it looks like, is if you have a mostly red spot, you will see mostly higher in the tumor, you'll see a red spot on the array. If it's mostly in the sample, you'll see a green. And if they're both hybridizing, you'll see different shades of yellow depending on the relative proportions of the two. So you're always looking at relative proportions in this particular type of RNA. So they are labeled red or green. Sometimes people do it both ways just to make sure you get the same answer both times. So these fluorescent dyes produce the different color signals when they're activated by a laser. Then they're scanned and then further analyzed. So I think I just said this. The appearance of the spots tells you which hybridized better, the tumor sample or the reference sample. And it's actually the ratio of the two signals, tumor versus reference, tells you whether the tumor expressed more, less, or the same amount of that particular RNA. So the data processing here goes on. What you are shown is never actually the array itself. They're scanned, a lot of calculations are done, and the sample colors have to be converted into some kind of fold change that is a number, and then that will be expressed or shown as red versus green or blue versus yellow more commonly now, as a reflection of the overall induction or repression of each RNA. So what you see is a very intensely calculated color, which is not the original color that you see on the array. Okay, so this is not a Mammaprint array, but it gives you the idea what's going on. Each column is a tumor, and the tumors are clustered into what types they are according to what cell types are involved. So we've got basal B, basal A, and luminal. So we're going to group the tumors according to what cell types are involved. And then we're going to look at all those tumors. Each row is a gene to see whether that particular gene is overexpressed or underexpressed in that particular tumor versus a reference. So you can see that the tumors do cluster. There's a whole bunch of genes here up at the top where the basal B cells have a very high expression level of this set of genes. And similarly, on the other end of the thing, the luminal have a very overexpressed group of genes on the lower right. So the tumors can be clustered not only according to their cell types, but according to what genes are over and underexpressed in those tumors. So that's the type of data that's used for all these types of arrays. And the genes that were chosen were very characteristic of different types of tumors to develop this 70 gene array, which is a really relatively small number of genes actually. But this gets across the idea that different tumors are different in what genes they are over and underexpressing them, and therefore if you know what genes to expect to be over and underexpressed, you can predict what kind of tumor it is for, in this Mammaprint instant, how that tumor is actually going to behave in the future. This is what I just said. Each row represents a gene. The color reflects its relative expression level. Each column is a tumor, and they're grouped to emphasize different gene expression patterns, which are characteristic of the specific types of tumors. And that's what's important. The different types of tumors do have different gene expression patterns, and that those can be used predictively. Okay, so in developing this test, the people had a very large number of tumor samples from women who'd had their cancers diagnosed a few years earlier, so we knew what the outcomes were going to be. So we already knew, they knew, which tumors had a poor prognosis that led to distant metastasis within 5 years, and they knew which ones had good prognosis. So what they did was characterize the expression patterns of a lot of genes in those tumors that early on to try to group them according to which ones were predictive of poor prognosis versus good, based on the expression levels of the RNA, the patterns of expression levels. Okay, so what's done now is the 70 gene signature was developed by comparing these and choosing the RNAs that were most useful or most characteristic of either well-behaving, good prognosis, or poor prognosis tumors, retrospectively. So now women with early-stage breast cancer, their cancers can be compared essentially to these known outcome tumors, and their expression patterns can be compared, and that can be used as a prediction, a predictor of whether those tumors are going to be a problem later or not. Okay, so this is results from early work, and this is what it looks like in terms of the expression patterns and their prognosis defect of prognosis abilities. That in this case, each row is a tumor, and each column is a gene, and these gene expression patterns are grouped according to the good prognosis versus bad prognosis group. So the genes that correspond to the little white bars on the right-hand side, those are the bad prognosis tumors. So those tumors had a certain cluster of genes which are underexpressed characteristically, and some that are overexpressed. The little black bars indicate tumors with good prognosis, and those are, include a certain group of low expression genes and a few overexpression genes. So what's done basically now is to compare a new tumor to the expression patterns that we see here to try to decide if it merits a black bar, which is good, or a white bar, which is not good, in terms of the prognosis. So that's what's done now. A score is assigned as good or bad, and that can be used to guide clinical decisions. Right, so we're clustered similar expressed genes are put together, and the white bar indicates that the disease progressed to metastasis within 5 years, and the black bar indicates that the disease did not progress within 5 years. And so by comparing a new tumor to those tumors, we can make some prediction about whether that particular tumor is likely to progress or not. So they report it as a score, like a yes or no answer. But the idea, of course, is to guide, not necessarily determine, but guide whether chemotherapy is needed in that particular case. Okay, there are others that are similar. Something called Oncotype uses 21 genes. This is Mammaprint, it uses 70. Mammostat 5 and Prosigna 58. But the idea is the same. The expression signatures of the various genes are believed to predict the likelihood of recurrence and the need for radiation and or chemotherapy.